my colleague Keith Elkon and myself will be chairing this. Keith Elkon is a researcher. He's head of the Division of Rheumatology, Department of Immunology at the University of Washington in the state of Seattle in the US. And he will introduce our first speaker. Please.
Yeah. Good morning. The first speaker is Dr. Megan McKay, who is Associate Professor of Medicine at the new medical school called Hubscher School of Medicine, North Shore, Long Island Jewish Health System. She's a researcher in the Feinstein Institute and works with Betty Diamond. So she'll give the first lecture.
Morning. It's a pleasure to be here. And I wanted to thank the organizing committee for graciously extending the invitation to me to come and present this story on behalf of Dr. Diamond, who apologizes for not being able to be here this morning in Buenos Aires. So it is possible to meet 4 of the ACR criteria for diagnosis of systemic lupus in the absence of autoantibodies. But I think that everyone in this room would agree that autoantibodies really define this disease, and it's reasonable to consider that they may be contributing to pathogenesis of some of the neuropsychiatric disorders that we attribute to systemic lupus. So virtually all of lupus patients exhibit anti-nuclear antibodies, and anti-DNA antibodies are both diagnostic and pathogenic, either through immune complex deposition or binding to DNA in tissue or cross-reactivity with other tissue antigens. And indeed, 15 years ago in Betty's lab, it was determined that a subset of these anti-DNA antibodies cross-react with portions of the NMDA receptors and contribute to behavioral and cognitive deficits in mouse models of lupus, and as we are beginning to learn, probably also in humans with lupus. So the scope of the disease is large, but it's a little bit difficult to pinpoint exactly how much. Using the 1999 ACR criteria for neuropsychiatric lupus, different cohorts have published prevalences of neuropsychiatric disease anywhere from 37 to 95%, with varying prevalences of cognitive dysfunction and mood disorder. And more recently, Inception cohorts have published lower incidences of neuropsychiatric lupus around or at the time of diagnosis. In the SLIT cohort, it was, I believe, between 6 and 12%, and even lower in the brain connection cohort, with the exception of cognitive impairment. In the brain connection cohort, all of those subjects had neuropsychiatric testing at the time of enrollment, and a surprising 60% of them demonstrated mild cognitive impairment, and 19% demonstrated moderate cognitive impairment. So I think the impact of these cognitive difficulties is demonstrated pretty graphically in this slide. The Ray-Osterrieth test asks subjects to reproduce complicated figures, as the one shown at the top of this slide. First by copying it, which utilizes visual spatial abilities, and then through recall, which is a test primarily of working memory. And we can see very dramatic differences between control subjects and the lupus subjects, but there are also dramatic differences between these 2 representative lupus subjects. And we would postulate that the variability between these 2 representative subjects, lupus patients, may in fact be mediated by neurotoxic antibodies. So the story of these antibodies began with a search for different antigenic specificities for anti-DNA antibodies. And the R4A is a mouse monoclonal anti-double-stranded DNA binding antibody that was used to screen a phage library, and it bound to the consensus sequence of the Dewey's peptide that bound in the DNA binding site of this antibody, and in fact soluble forms of this Dewey's peptide inhibited binding of the antibody to both R4A and double-stranded DNA. And Chaim Putterman then went on to immunize non-autoimmune BALB/c mice with a multimeric form of the peptide on the upper right-hand corner of the slide, connected to a polylysine backbone. And these mice demonstrated an exuberant IgG anti-double-stranded DNA response that was not seen in the similar mice immunized with MAP-core peptide. And levels of these anti-DNA antibody titers were equivalent to those seen in NZW lupus-prone mice, and their pathogenicity is demonstrated on the lower right with glomerular binding. The same consensus sequence, the Dewey sequence, was also found to be located within the binding domains of the NR2A and NR2B subunits of mouse and human NMDA receptors. And these receptors are distributed throughout the brain with predominance in the hippocampus and amygdala, and they are located within glutamatergic synapses where they form biologic factors for learning and memory. And neurons communicate through synapses. So following electrical stimulation, the agonists glutamate and glycine are released, and they bind to, as you see in the top cartoon there, they bind to the subunits, opening the pore and allowing calcium entry into the cell. And prolonged opening of this pore or stimulation results in high calcium influx and death of the neurons. So initially, studies by DiGiorgio and others demonstrated that this cross-reactive R4A antibody binds to both the NR2A and B subunits in vitro. And injection of these antibodies in vivo into black 6 mice results in neuronal death. And you can see in the slides that there's disruption of the, there's disruption of the CA1 area of the hippocampus and dying neurons in the mice injected with R4A that's not seen in those injected with control antibody. And it was also established that these cross-reactive antibodies preferentially bind activated and open receptors, and that consequences of binding to these receptors is dependent on antibody concentration, such that low concentrations of antibody yield enhanced excitatory postsynaptic potentials and affect synaptic plasticity, whereas high levels will result in neuron death. So we have a clinical conundrum that in a healthy state, the blood-brain barrier prevents autobody access to the central nervous system. So how do these antibodies gain access? Because in fact, the immunized mice that have circulating neurotoxic antibodies are phenotypically normal until the blood-brain barrier is disrupted. And Chesha Kowal and Patricio Huerta demonstrated that not only does disruption of the blood-brain barrier allow antibody access with immediate loss of neurons, but the agents used to breach the blood-brain barrier confer regional specificity on these antibodies. So for example, in the top slide, the blood-brain barrier that's breached with lipopolysaccharide, these neurotoxic antibodies hone to the hippocampus, and the animals subsequently develop an impairment of their spatial memory where they can't navigate a water maze adequately. Conversely, the same animals that are exposed to epinephrine have a breach in the blood-brain barrier, and the antibodies hone to the amygdala. And although their spatial memory is intact, they no longer have an appropriate response to a fear conditioning paradigm. Now, spatial memory is a measurement of the environment and an individual's response to the environment. It relies on specific specialized cells within the hippocampus that are called place cells. And we know from recordings in mice that spatial information is encoded in the patterns of place cell firing, so that as animals that have these electrodes implanted in them, as they traverse a place field, such as this mice is doing, as they approach certain areas, individual place cells will fire. And the collective firing of multiple place cells gives rise to these specific spatial maps that you see here. And NMDA receptors are part of the process in the development of the specificity of place cells. So Eric Chang utilized the immunized mice to determine the consequences of these anti-NMDAR antibodies on place cell firing. And you see that initially in both sets, the MAP-DUIs and the MAP-core immunized mice, the place cell fields are identical. However, following lipopolysaccharide administration, the place field in the MAP-DUIs immunized mice, where the neurotoxic antibodies gain access to the brain, are suddenly dramatically increased in size, as shown in the bar graph on the right. And we can look at this in a slightly different way, where spatial information is an actual measurement of the amount of firing of these place cells, and we see that this decreases in the animals immunized with Dewey's peptide and exposed to LPS. And this gives rise to the increased size of the place cell that you saw just a minute ago. Now, these effects are long-lasting. They last at least 9 weeks after exposure to LPS, even though the antibody can no longer be detected 2 weeks after exposure to LPS. And this suggests that persistent cognitive deficits do not rely on constant exposure to this antibody. And Bruce Volpe then looked at the remaining neurons in the hippocampus of these mice, and he focused specifically on the dendritic processes of neurons, because it's known that decreases in numbers of dendritic processes associate with impaired memory. And indeed, he found that in the MAP-DUIs immunized mice, after exposure to LPS, there's a significant reduction in the number of dendritic spines compared to the intact architecture of the neurons in the MAP-core immunized mice. So this is a structural representation, perhaps, or explanation of the deficits in the spatial memory in these mice. And we postulate that this may occur because of a process called, I'm forgetting the name. I knew I'd forget the name. It's glial cells that migrate and phagocytize these dendritic processes. And this occurs normally in embryonic development in a process called synaptic pruning. In our model, we postulate that exposure to neurotoxic antibody results in death of neurons, and this process recruits the glial cells to the damaged area where they subsequently might phagocytize these remaining neurons. So Betty's lab then very recently looked at the fate of these glial cells within the hippocampus in the mice that have been immunized and treated with LPS. And on the left, the glial cells are stained with green, and on the left we see normal architecture in unmanipulated mice, which is mildly disrupted in the control immunized mice exposed to LPS. But in the peptide immunized mice on the right, it's drastically disrupted, and the glial cells are really cheek to cheek with the remaining neurons, suggesting that they could have migrated to this area and might be involved in the phagocytic process of the dendritic spines. We've also learned a great deal about neuronal death and compensatory responses through evaluation of resting metabolic activity in the mouse model. So we used micro-FDG PET studies to measure glucose metabolism in the resting state. And prior to LPS, we can see that both sets of immunized mice with MAP-DUIs and MAP-core have similar amounts of metabolic activity. Following 2 weeks after LPS administration, this activity drops in the MAP-DUIs immunized mice, which correlates with the loss of neurons in the hippocampus. It then increases by 4 weeks. And conversely, in the control mice, we see an elevation of metabolic activity at 2 weeks, which begins to level off at 4 weeks. And we think that this is associated with a non-specific microglial response to the LPS insult to the blood-brain barrier. But if we look at the rate of metabolic increase from 2 weeks to 4 weeks in both sets, we see that the rate of increase in the Dewey's immunized mice is twice that of the MAP-core immunized mice. And we postulate that this is part of a compensatory response, either by the remaining neurons or by the glial cells supporting those remaining neurons. And looking at the histology in the brains of these mice at 4 weeks, sacrificed at 4 weeks, we see a significantly decreased number of neurons in the hippocampus of the peptide, Dewey's peptide immunized mice compared to the control mice. And there's also a marked, significantly inverse correlation between metabolic activity and neuron number in this region, again suggesting that increased metabolic activity with a lower amount of cells is representative either of neural compensation or glial compensation for the neurotoxic insult. So what we've learned from these mouse models is that both cognitive and behavioral disorders may be mediated by antibodies, and there may be many brain-reactive antibodies. The same antibodies can produce more than one clinical syndrome, and agents that compromise the integrity of the blood-brain barrier expose neurons to these antibodies with regional specificity, again causing different clinical syndromes, and that glial cells may compensate for neuronal dysfunction. So moving to the realm of human lupus, the story is not quite so clear for many reasons. So in patients, the anti-NMDR antibodies do not exist as positive or negative, but rather as in a continuum, and we expect that the results of this would be variable as well. We also have no way of predicting how and when these antibodies would gain access to the central nervous system. And current measures of cognitive function and behavioral function are imprecise, and they are frequently confounded by other variables such as disease activity, medications, hormonal and metabolic disturbances, and a variety of other things. And we think that these are some of the reasons why at least 5 studies have failed to appreciate significant correlations between serum levels of autoantibody and cognitive dysfunction. However, 3 studies have demonstrated correlations between CSF levels, spinal fluid levels of the neurotoxic antibodies, and central nervous system manifestations of lupus. One of these studies is shown here in this slide from Fragoso Loyal and others in Mexico, where the spinal fluid had markedly elevated titers of the anti-NMDR antibodies in patients with central nervous system manifestations such as acute confusional state, severe cognitive dysfunction and seizures, compared to those with peripheral neuropathies or patients undergoing surgical procedures. Importantly, the levels of anti-NMDR antibodies in these patients were sufficient to cause neuronal death in vitro. And it was also shown in this study that 6 months after the initial presentation, many of these patients had second lumbar punctures. And although many of the pro-inflammatory cytokines that were demonstrated at the initial presentation remained elevated, the anti-NMDR antibodies were no longer measurable, which is analogous to the mouse model where these antibodies were not measurable 2 weeks after LPS administration. So because in the mouse model it was demonstrated that the antibodies affect the place cells with effects on spatial memory, we employed specific memory testing in a cohort of lupus patients stratified by presence or absence of these anti-NMDR antibodies and matched healthy controls that tested both non-spatial and spatial components. And the assay is shown on the left-hand side of the slide. The subjects were shown an array of 4 objects for a couple of seconds, and then it was removed. They were then asked whether a particular object was present or absent, which is non-spatial memory, and then they were asked whether that object was above or below or on the right side or left side of another object, which is spatial memory. And the bar graphs on the right, the healthy controls are in blue, The anti-NMDAR positive lupus subjects are in tan, and the negative ones are in green. And we see that on the non-spatial testing, all groups performed well and equivocally on the non-spatial testing. However, in the spatial testing, all groups did significantly worse, and in fact there were significant differences between healthy controls and lupus subjects, and between the healthy controls and lupus subjects with circulating anti-NMDR antibodies. So this demonstrates that seropositivity for these antibodies correlates with spatial memory impairment in lupus patients as seen in the mouse model. We also recently completed analyses of studies looking at resting brain metabolism in lupus patients compared to healthy controls, and discovered that lupus patients have significantly increased resting metabolism in the hippocampus, orbital frontal cortex, and putamen compared to the healthy controls. All of these patients had no evidence of neuropsychiatric involvement before or during testing, and at the time of testing, all of them had stable disease and stable medications. None of the clinical assessments of these patients, including things such as disease duration, age, ethnicity, complement levels, other antibody levels, correlated with the hippocampus hypermetabolism. However, increased levels of serum anti-NMDAR antibodies and increased hippocampal metabolism correlated independently with decreased performance on memory testing. We utilized ANAM metrics for this study. And regression analysis demonstrated that the combined combination of these two variables predicted a worse functioning on these memory tests compared to either one alone. So we now have a possible situation where lupus patients may exhibit increased metabolism in the hippocampus without memory impairment, as we had seen in our mouse model, but memory impairment or clinical dysfunction will rely on the presence of a neurotoxic antibody. So thinking about these antibodies and how they get through the barrier and where do we not have a blood-brain barrier leads to the idea to placental transfer. And it's been known for a long time that children of lupus moms are at increased risk for learning disabilities. And we know that placentas readily traverse that antibodies readily traverse the placenta, and harmful effects of lupus antibodies have clearly been demonstrated by Jill Byon with the anti-Ro antibodies. So it was reasonable to ask the question whether these cross-reactive anti-NMDR antibodies can cause neuronal damage in the developing fetus that does not have an intact barrier. The experimental design was to immunize female mice prior to mating and pregnancy. Some of them were sacrificed during embryogenesis to look at fetal brains, and all of the offspring were assessed with different behavioral assessments and sacrificed at 8 months for brain histology. And looking at fetal brains at embryonic day 15, we see that tunnel staining shows that exposure to maternal anti-NMDAR antibody in utero results in greatly increased numbers of dying neurons on cortical slices. And these same antibodies again cause cell death and abnormalities in the developing fetal neocortex. Here we see the cortical plate, and in the control immunized mice, the cortical plate looks nice and healthy with the DAPI staining. But in the NMDAR exposed antibody mice, it's very thin and kind of moth-eaten, and the cortical plate to cortical width ratio is much lower in the antibody exposed mice. To prove that the anti-NMDAR antibodies were indeed responsible for this phenomenon, non-immunized pregnant mice were passively given the R4A cross-reactive antibody and its human counterpart, the G11 antibody, that also binds double-stranded DNA and NMDAR antibodies. And again, it was shown even 3 days after administration of these antibodies, that mice exposed to them in utero developed the problems with cortical plate development that was not seen in the control antibody injected mice. And the offspring of these mice also show impaired behavioral performance on specific tasks. So normally, a mouse that looks at a novel object will explore that preferentially. And this normal behavior was not demonstrated in the mice who had been exposed to this antibody in utero, and nor did they recognize transposed configurations of objects. So here we have a story where these neurotoxic antibodies cross the placenta. They cause brain defects in developing mice that are translated into long-term cognitive deficits. And it was also noted during these studies that the the peptide immunized mice consistently gave birth to more males than females. And indeed, the Dewey's peptide immunized mice give birth to 31% of females compared to the control immunized mice of 51% female, which is a markedly decreased female-to-male ratio. And looking at histology of the fetuses in the MAP Dewey's immunized mice, there are no gender differences in any of the internal organs. However, there were markedly increased numbers of degenerating and dying neurons in the brainstem of the female fetuses. And this was related to enhanced NR2A expression in the brainstem of these female fetuses. So NR2A knockout mice were then immunized again with the MAP Dewey's and MAP core peptides, and no gender differences were observed in the offspring, demonstrating that the female fetuses are viable in the absence of NR2A subunits. So we have seen that brain-reactive antibodies then can alter fetal brain development when there's an incompetent blood-brain barrier, and it can cause sex ratio skewing in mice with significant loss of female fetuses. So just lastly to mention, how can this knowledge translate to benefits for our patients? And we've known for a long time that the D-peptide of the Dewey's, the configuration of the Dewey's peptide protects neurons. So the top two panels demonstrate amygdala slices in Dewey's immunized mice exposed to epinephrine and treated either with D-peptide or saline. And you can see that the neurons in the D-peptide-treated mice look much healthier than the dying ones in the saline-treated mice. And similarly, in the mothers that have circulating anti-NMDR antibodies, the only fetuses who have normal cortical development are those whose mothers were treated with the D-peptide isomer of the Dewey's, and not those treated with irrelevant peptide or saline. So Ona Bloom and Youssef Alabed at the Institute have collaborated. They've used features of the Dewey's peptide to create a small molecule peptide mimetope called Fissile 412. And this molecule very effectively abrogates binding of these cross-reactive antibodies to tissue substrate. And you can see in the bottom panels that it inhibits binding of the cross-reactive R4A and human G11 autoantibodies to DNA much more effectively than the Dewey's peptide. And if this compound is also given with the R4A and G11 antibody injections into brain, it confers neuronal protection. So we have demonstrated that these mouse models and the human studies work together to demonstrate that brain-reactive antibodies may arise as part of an autoimmune or protective immune response and may contribute to cognitive or emotional impairment. And maternal antibodies may contribute also to abnormal fetal brain development and persistent cognitive deficits. And decoy antigens may represent a therapeutic strategy. Thank you for your attention. I'm happy to take questions.
Thank you so much. What shall we do, Keith? Shall we move on? Because, you know, see the time there?
Oh, over time.
Maybe just one question there. No?
We have time for 2 questions, please. Go ahead.
Can't see.
Can I just ask a quick question about the role of cell, over here, to your right. There you go.
Oh, thank you.
About the role of the neuronal cell death versus cell dysfunction or NMDA dysfunction. Do you have to have hippocampal cell death to see the impairments that you're getting, or could you simply get these impairments with disruption of NMDA functioning? Because I'm thinking about in the context of patients where if it does require the death, then it would suggest that the dysfunction, in fact, would be irreparable.
No, you are correct that actually these antibodies can mediate both transient neuronal dysfunction if they are low-level concentrations, and they mediate neuronal death at high concentrations. And this can mimic what we do see clinically, which is that some of these clinical syndromes are transient and some are fixed deficits that occur incrementally over time.
Since the data are not so clear in humans, how specific are the antibodies there? Or could they be part of the polyreactive antibodies? Or is this clear?
I'm sorry, I didn't.
So you said that a number of studies could not really show a correlation between the antibodies. And is there a possibility that some of those antibodies which are fluctuating could be part of polyreactive antibodies? Or are they very specific?
They are specific. It is possible that they could be fluctuating at different points in time. But remember, these studies are all done with serum antibodies. And again, we have no way of knowing when and how they're going to access the central nervous system. And that if you look at cerebral spinal fluid, the correlations are much more clear. The problem is we can't access spinal fluid very easily. And that's why, again, I think the pursuit of neuroimaging studies may in fact be the way that we're going to be able to diagnose these. If we can correlate specific changes on neuroimaging studies with the presence of antibodies, we'll have a way to detect progression of disease that's related to these antibodies.
Very good. Thank you very much, Doctor.